Давайте. И что, я могу начинать читать? Хорошо, спасибо. Я начинаю. Мгм. Спасибо. Uh, Good afternoon, ladies and gentlemen. Today I take the first lecture in microbiology. Topic of today's lecture is morphology, structure, and physiology of bacteria. This is a very large topic. I hope that I will comment at least most important things. Microbiology is the science of microorganisms. Microorganisms, also named as the microbes, are the minute organisms invisible to the naked eye. It means all the organisms that may be seen in the microscope may be named microorganisms. All the microorganisms may be subdivided into several groups. These groups are subdivided into large subgroups. First, cellular entities of microorganisms. Cellular entities, they are microbes that have cellular structure, cells. Among them are bacteria, this is the topic of today's lecture, fungi. In the context of bacteria, fungi eukaryotes, bacteria prokaryotes, and protozoa. What in the slide here you see two cells of the Trypanosoma vaginalis. They are cellular entities among the microbes. Non-cellular entities, second subgroup, or subcellular or non-cellular entities. Among them, first of all, of course, are viruses. Viruses are not cells, and virus unit that has a certain shape, infectivity, and other usually is named by the word particle. Virus particle or virion. Second type of subcellular non-cellular entities are prions. Prions are infectious proteins. They don't contain nucleic acids. And the last entity is named viroid. Viroids are ribonucleic acids. Free ribonucleic acids are not coated by the special protective membranes. Viroids cause diseases in plants usually, not in humans. Size of microorganisms. Microorganisms, their size vary in the spectrum. Here, what in this figure you see spectrum from 1 millimeter to angstroms. Prokaryotes, shown here the yellow sector, have size from 0.23 micrometers to 3 to 5 micrometers. Larger organisms are fungi. They have size from 3 to 10 micrometers, and some of them are larger. Viruses, what here in this column you see, this is a green sector. Viruses usually are measured in the nanometers. No, 1 micrometer, this is 1000 nanometers. Our today's topic is bacteria, prokaryotes. Let's compare major structural characteristics of bacteria and fungi. Bacteria as prokaryotes and fungi as eukaryotes. They have some similarities and some differences. Prokaryotes, bacteria, DNA. They have DNA in the form of the secular double-stranded DNA chromosome. Majority of bacteria have only one chromosome, but some of them may have more than one. Fungi have their DNA in the paired chromosomes. Localization of nucleic acid. Nucleic acid in prokaryotic bacterial cells occur in the so-called nucleoid. Nucleoid, this is a region of the cytoplasm contain DNA chromosome of the bacterium. Nucleoid is not surrounded by the nuclear membrane. This is a place in the cytoplasm. Contain DNA that is not surrounded by a special membrane. In the contrast, in fungal cells, DNA is localized in the true nucleus. This is a special organism surrounded by the nuclear membrane, and all the chromosomes occur inside of this. Nucleoli. Bacteria don't have nucleus and they don't have nuclei. Fungi, eukaryotes, of course, in the nuclei present. Histones. Histones are the basic proteins that intimately interact with the nucleic acids, usually in the fungi and other eukaryotic cells, in eukaryotes. Histones in the bacteria are absent. Endoplasmic reticulum and Golgi apparatus. In the bacterial cells, these membranous structures are absent. In the fungal cells, of course, they are present. Plasma membrane. Plasma membrane of bacteria contains different substances, but they don't have sterols. It means plasma membrane of the prokaryotic bacterial cells don't supplemented with sterols. Eukaryotic cells of fungi and other eukaryotes, these membranes contain sterols. Now, this is a basic biochemical difference between non-sterols in the bacteria and presence of sterols in the eukaryotes. And finally, cell wall. By the way, cell wall, this is a coat of the cell external to the plasma membrane and responsible for rigidity of cell and shape of cell. Also, cell wall protects bacteria from the osmosis, osmotic pressure. So, in the bacterial cells, cell wall consists of the peptidoglycan. This is a rigid backbone of the cell wall. In the fungal cells and our cells and cells of other eukaryotes, peptidoglycan is absent. Cell wall maintains its resistance due to presence of the cellulose and chitin. Chitin, you know that chitin is very typical for insects. When saying about major steps of the development of the microbiology, of course, we have to remember names of the three famous scientists. First is Anton van Leeuwenhoek. In the 1660s years, Leeuwenhoek first discovered microbes because created the first microscope that allowed him to see these very, very small animals. Second scientist is Louis Pasteur, French scientist, famous in the 1860s years. Louis Pasteur elaborated the technology of the production of vaccines. Vaccines are the medical preparations that are used to prevent infectious diseases. But author of the vaccine technology is Louis Pasteur. Robert Koch, third scientist, German scientist. In 80s, 70s years, Robert Koch discovered causative agents of two diseases, of the cholera and of the tuberculosis. Robert Koch is a Nobel Prize winner for the discovery of many things concerning tuberculosis. Robert Koch, for example, elaborated so-called tuberculin that now is named as the PPD. This is a allergen that is used as a diagnostic reagent for study immunity against tuberculosis. From this slide, we will say only about prokaryotic bacterial cells. You see sector that is occupied by prokaryotes in the scale of the sizes, and see that prokaryotic bacterial cells have the diameter of 0.2 to 1.5 micrometers, and if they are rod-shaped bacteria, they have from 3 to 5 micrometers long. Very important characteristic of bacteria that is seen in the microscope is the shape and arrangement of bacteria. On the basis of the shape, bacteria are subdivided to the bacilli, the rod-shaped bacteria, cocci, round bacteria, spirochetes, and spirilla. Spirochetes have the tiny coiled bodies, snake-like. Vibrios have the comma-shaped bodies. About arrangement. If round cells arrange in chain, they are named streptococci. If bacilli, rod-shaped bacteria, is arranged in chain, they are named streptobacilli. Strepta means chain. Staphylococci, they are round cells that form the clusters, grapes-like clusters. And diplococci, they're coupled cells when two cells bound one to other, forming all these couples. In this slide, you see photograph of the pus discharged from the urethra of the patient suffering from gonorrhea. This is sexually transmitted disease. In this slide, you see large pink objects. They are human leukocytes and very small dark red paired cells. They are diplococci. In the right part of the slide, you see what this couple of cells when one cell intimately contact to the neighboring cell, forming double cellular structure. This is a typical characteristic of the Neisseria. For example, Neisseria gonorrhea. This slide demonstrates you streptococci. Strepta means chains, what you see chains in the left-hand part photographs, and really what here shows Streptococcus pyogenes. Streptococcus pyogenes, genus name Streptococcus means that all the members of this genus would be round and form chains. Staphylococci. Staphylococci, you know, they are gram-positive. What after the first class, you have to remember that gram-positive means blue after gram stain. But gram-positive round cells that arrange in the grapes-like clusters. The most pathogenic, it means most dangerous in the plane of the causing of the human disease, Staphylococcus, species of Staphylococci, is named Staphylococcus aureus. This is genus name and species name. Species name consists of two parts. First epithet is a characteristic of genus, and second epithet is a characteristic of species, Staphylococcus aureus. Importance of this species is due to very high resistance for antibacterial drugs. Antibiotic resistance of Staphylococci really isolated from the patients all over the world. Rod-shaped bacteria. This slide contains two photographs. Photograph A demonstrates gram-negative, scattered, it means arranged atypically, rod-shaped bacterium that is named Escherichia coli. Escherichia coli is gram-negative. It means after gram stain, Escherichia develop pink color. In the right part, you see photograph B. Photograph B demonstrates appearance, microscopic appearance of Clostridium perfringens. Clostridium perfringens, in the contrast to Escherichia, is Clostridium, is gram-positive bacterium, dark blue. This also atypically arranged cell. By the way, Clostridium perfringens causes gas gangrene. This is a severe infection that is developed after trauma. This slide demonstrates rod-shaped bacteria. Photograph A demonstrates small rod-shaped bacteria, some of them arranged in couples. Here you see that these bacteria are surrounded by white, it means colorless capsules. They're encapsulated rod-shaped bacteria. Species is named Klebsiella pneumonia. Photograph B below the A demonstrates Bacillus anthracis, streptobacilli. Gram-positive means blue color. Strepta chain-forming organism. Bacillus anthracis causes severe disease that is named anthrax, and naturally this disease is acquired after contact to animals. The next slide demonstrates shape of spirochetes. Spirochetes are spiral-shaped slender microorganisms, usually motile. Photograph A, that has a green background, demonstrates Treponema pallidum. Treponema, here you see 4 cells, 1, 2, 3, 4. Of Treponema pallidum, they have hooks on the bodies, and you know maybe that Treponema pallidum is the causative agent of the syphilis. This is a sexually transmitted disease. In the photograph B, you see other spirochetes, more rigid, less flexible. They are spirilla. Really, you see Rhodospiridium rubrum. Spirilla usually are another important causative agents of human diseases. Structure of bacteria. We told you about tradition of the subdividing of all bacteria to the gram-positive and gram-negative organisms. After gram staining, all the gram-positive bacteria develop blue color, gram-negative pink. In this scheme, you see cutted bodies of the gram-negative bacterial cell in the left part of the painting, and this is gram-positive on the left and gram-negative in the right part of this painting. Now, first of all, take a look to the center. In the center, you see brown large molecule. This is a DNA chromosome of the bacteria that is situated in the cytoplasm of the gram-positive and gram-negative bacteria. You see that chromosome in both these cells is not surrounded by the nuclear membrane, and due to this we say that these cells are prokaryotes. Nucleoids, so a nucleoid, this is a part of the cytoplasm, contain DNA, genome of the bacteria. What central part of this structure is named nucleoid because it consists of the DNA. DNA is surrounded by cytoplasm. What here? Cytoplasm surrounds the DNA, and here. Cytoplasm, this is a colloid system, contain such organelles as ribosomes. Ribosomes is both gram-positive, gram-negative cells, what in this figure shown as the small rings. Exception for ribosomes, here occur inclusion bodies. They are typical for both gram-positive and gram-negative cells. Inclusion bodies, they are storages of the nutrients that cell accumulate in the cytoplasm when actively metabolized substrates. Cytoplasm is surrounded by the plasma membrane. Plasma membrane is shown as a thin blue coat surrounding cytoplasm of the both gram-positive and what now I demonstrate gram-negative organisms. In some cases, cell membranes of the bacteria form the invaginations of cell membrane inside of the that are protruded to the cytoplasm. Well, these invaginations of the cell membrane are known mesosomes. What here you see one invagination, mesosome, in gram-positive bacteria. Mesosomes usually form cross walls between two daughter cells in the process of the dividing of maternal cell into two daughter cells. What beginning of the formation of cross walls between two neighboring daughter cells consist of the mesosomes initially. Plasma membrane is surrounded by the cell wall. The gram-negative bacterium cell wall is shown as the purple protective layer surrounding plasma membrane. And you see that this is a rather simple structure. But in the gram-negative bacteria, cell wall is more complex and consists of the peptidoglycan layer, what stripe structure in the right cell, gram-negative cell. This is a peptide-glycan layer. And the cell wall of gram-negative bacterium contains additionally outer layer. What this thin layer with a bubbles inside? This is the outer layer of the cell wall of the gram-negative bacteria. It means cell wall of gram-negative bacteria consists of two elements: internally localized peptide-glycan, externally localized outer membrane. In the gram-positive cell wall, only peptide-glycan layer. Cell wall is surrounded by the capsule. Capsule is shown here, the external protective layer of the gram-positive bacteria and external protective layer of gram-negative. Exception for these structures, some structures of bacteria are protruded outside of the bacterial cells. First of all, they are flagella. What is the left flagellum? Brown filament, and the brown filament analogous in the gram-negative bacteria. Flagella contain consists of the flagellin. This is a contractile protein that responsible for movement of cell, propels the cells to the certain direction movement. Some cells are not flagellated and they are not motile. In the scheme that demonstrates gram-negative bacteria, in the lower and other parts you see short rod-shaped structures that penetrate capsule, but they are shorter than flagellum. They're so-called pili. Pili are filamentous proteinaceous substances that are responsible for attachment of bacteria to the substrate, to the other bacterial cells, or to human cells, and serve as the pili serve as the attachment instruments of bacteria. Now, let's go. Briefly, let's characterize different structures of bacteria. First of all, cytoplasm. What here cytoplasm is shown as the yellow material inside of the bacterial cell contain red ribosomes. So cytoplasm is the colloid system of a variety of organic and inorganic solids in the viscous water solution. It means cytoplasm is a viscous material. Cytoplasm may be subdivided into distinct areas. First is amorphous matrix. This is a major part of the cytoplasm. This amorphous matrix contains metabolites, ribosomes, mesosomes. What, by the way, what mesosome is shown here, the invaginate plasma membrane to the cytoplasm. Inclusion bodies, nutrient granules, vacuoles, plasmids, and some other structures. In the contrast, second compartment of the cytoplasm is named nucleate region. Nucleate region here is shown as a large brown belt-like structure consisting of DNA. It means nucleoid. This is the part of the cytoplasm where nucleoid is present now. Cytoplasmic membrane. Cytoplasmic membrane surrounds the cytoplasm, consists of the phospholipids. This is a bilayer of phospholipids, and the proteins integrated inside or on the surface of this lipid bilayer. Functions of the plasma membrane. First, this is an active transport of metabolites into the cell or outside from the cell to the from our cell to the outside. Active transport is realized by the presence of the porine proteins that form the specialized tube-like protein structures that are specific for substances. Some of them specific substances that are transported from outside to inside of the cytoplasm. They're usually nutrients. Some of them are responsible for releasing of the substances produced inside of bacteria to the outside, for example, exotoxins. Second function of the cytoplasmic membrane, generation of energy. Some of the proteins integrated to the plasma membrane are flavoproteins and other substances, cytochromes, for example, that are included in the process of the transport of the electrons from one the integrated plasma membrane-associated protein to other. Well, this electron transport is required to generate energy because energy is accumulated in the form of the protons accumulated in certain area of the cells or other ions, and typical place of the conserving of the energy. This is adenosine triphosphate that is situated at the cytoplasm below the plasma membrane. Third function, synthesis of cell wall precursors. Plasma membrane contains some of the enzymes that are responsible for synthesis of the peptidoglycan, for example. And last function, secretion of enzymes and toxins. Through the porine proteins, some enzymes produce the bacterial toxins may be released to the outside. Mesosomes. You know that mesosomes, what here, or here, mesosomes, are the coil-invaginated cytoplasmic membranes. What's the right photograph? You see this very good. These invaginated cytoplasmic membranes, invaginated to the cytoplasm, have the following functions. They serve as the origin of the transverse septum that divides the cell in half in the process of replication. Mesosomes are invaginated to the cytoplasm from one side and opposite side of the cell. And finally, mesosome 1 and second types fuse one to other, forming cross-wall. What in the photograph on the left upper part of the slide, you see two daughter cells immediately after formation of this transverse septum that separates one daughter cell from other. Second function of the mesosome, it pulls apart daughter chromosomes during cell division. It means mesosomes are responsible for separation of molecules DNA that has to be transported to both daughter cells, upper and lower cell. And this is also function of the mesosomes. And the third function, mesosomes participate in electron transport and energy production. Electron transport and energy production. We know that plasma membrane contains proteins of the system of respiration that are responsible for energy production in the cell. Mesosomes are invaginations that enlarge the surface of the plasma membrane, and in this way, mesosomes give additional energy to the cell. This energy becomes very important in the process of division of the cell. The next structure, nucleoid. We know that nucleoid is the chromosomal DNA situated in a certain place of the cytoplasm. Nucleoid is the bacterial genome, located in the form of the haploid bacterial chromosome. Genome consists of the single circular molecule of the double-stranded DNA. Single molecule of double-stranded DNA that usually has the circular structure for majority, not all, for majority of cells. Molecular weight in daltons of the average size of the chromosome of the bacterium is about 2000 million dalton. 2000 million dalton. This size is enough to form about 2,000 genes. No, one gene, this is a sequence of nucleotides responsible that codes for one polypeptide, functionally active polypeptide. What bacterial cells have genomes about 2,000 genes are in the contrast human genome consists of about 100,000 genes. The nucleus of bacteria contains no nuclear membrane, no nucleoli, no mitotic spindle, no histones, and no introns. But they are characteristic of the a very brief, primitive characteristic of the bacterial genome. Exception for bacterial chromosomes, what in this photograph chromosome is shown as the ring yellow molecule. Exception for the chromosomes in many bacteria may be recognized smaller DNA molecules that are named plasmids. So plasmids are the extra chromosomal, it means that may situate separately from chromosome, extra chromosomal double-stranded circular DNA molecules, which can occur in both gram-positive and gram-negative bacteria. Plasmids, here is also plasmid, are capable of replicating independently of the bacterial chromosome. It means if bacterial chromosome usually replicates before beginning of the replication of the bacterium, in the contrast, plasmids may replicate separately, and number of the plasmids in the cell may be much higher than number of the chromosome molecules. Then, plasmids usually are extrachromosomal, how it's shown here, but in some situations, plasmids can be integrated into the bacterial chromosome. It means DNA plasmid may be fused to the DNA of the bacterial chromosome. In this case, we can say about integrative plasmids. And the last, plasmids often provide a selective advantage such as the resistance to antibacterial drugs. Plasmids are not essential parts of the cell. Cell may survive without plasmids, but presence of some of the plasmids provide advantages, preferences for the cells that have these plasmids. We know that plasmids is the DNA codes for protein. Some proteins may cleave antibiotics, for example, penicillin, and in this way protecting bacteria from the activity of this antibiotic. Ribosomes. Bacterial ribosomes are situated in the cytoplasm. They are rather small organelles consisting of two subunits. Bacterial ribosomes have the size of 70S, 70 Sverberg. This ribosome consists of two subunits, 50S, 50 Sverberg, and 30S, smaller subunits. In the cleft between the subunits usually is bounded messenger RNA that codes for proteins. And the ribosome serves as the factory producing polypeptide chains that are complementary to the structure of the protein encoded to the messenger RNA. Eukaryotic, human and fungal and protozoa organisms, ribosomes are larger. They have size about 80 Svedberg, 80S, and bacteria 70S. Granules. Some bacteria may accumulate nutrients or products of their metabolism very intensively in the cytoplasm, in different parts of the cytoplasm. Well, these storages for the nutrients may be visualized by special methods of staining. But here you see two photographs with the different methods of staining of the Corynebacterium diphtheria. In the low part, you see that bacteria have the blue color because in the low photograph we see a result of the stain of the Corynebacteria by the metachromatic methylene blue dye. Methylene blue dye stains bacterial cells blue, but what does it metachromatic? It means the same methylene blue stains volutin. This is a polymetaphosphate accumulated in the bacterial cells, stain into the red color. It means cytoplasm is stained by methylene blue. The blue part of the bacteria, but on the periphery you see red dots. They are storages of the volutin that are stained by methylene blue, stained as the red ends of the bacteria. And now about cell wall. Cell wall is a very important structure responsible for gram stain of the bacteria. Cell wall is what here shown is the blue, here is the red material, is a multi-layered rigid structure external to the cytoplasmic membrane. External to the cytoplasmic membrane, what now? Blue. The basic structure of the prokaryotic cell wall is called peptidoglycan or murin. They're synonyms. In the gram-positive bacteria, murin forms the many layers of the polymerized murin subunits and form very rigid, strong cell wall of the gram-positive bacteria. They shown on the right. In the gram-negative bacteria, cell wall has two layers. Murin layer that is shown red, but in the gram-negative bacteria, this layer is very thin, small. And outer membrane that's situated external to the murin layer. Outer membrane consists of the phospholipids and the lipopolysaccharides. Gram stain is known from 1884 years. 1884 year, this is the year of the discovery of the gram stain method by the Swedish scientist Christian Gram. On the basis of the gram stain, all the bacteria subdivided into the gram-positive bacteria that develop blue color and gram-negative bacteria that develop pink color. You understand that gram stain, this is a method, complex method of stain. Complex means that two or more number of dyes are taken. Now you see that if gram-positive bacteria blue or gram-negative or pink, two dyes must be taken. It means Hansen valid blue dye for stain of gram-positive bacteria and Fuchsin that makes gram-negative bacteria red. In this slide you see structure of the cell wall of gram-positive bacteria. We see that cell wall of gram-positive bacteria consists of the parallel lines of the polymerized muramic acid and glucosamine. Muramic acid shown red, glucosamine yellow, and they are cross-linked in two directions. What glucosamine to the muramic acid along one line of the polymer, and the glucosamine molecules of the neighboring lines of the murin may be cross-linked one to other, forming very rigid structure. Exception for glucosamine and muramic acid in the structure of gram-positive bacteria, we can see cylindrical, filamentous, long molecules that are named teichoic acids. These teichoic acids may serve as the simple teichoic acids that are situated in the inside of the structure of the murine and the protrude outside, or lipoteichoic acid. What lipoteichoic acid is shown here. And here, lipoteichoic acids anchored to the phospholipid layer of the plasma membrane, what lower layer of this structure that you see is plasma membrane, and teichoic acids anchored to the plasma membrane penetrate murine layer, bridging plasma membrane to the murine layer of the gram-positive bacteria. Here you see gram-negative cell wall. Gram-negative bacteria contains murine. This murine layer is shown as the green, two lines of green myrmic acids and glucosamine, myrmic acid glucosamine. Also you see external to the murine layer, outer layer of the plasma membrane consisting of the phospholipids, they are shown yellow, and the lipopolysaccharides, they are shown also yellow and mounted to the external layer of phospholipids. Also outer membrane and of course plasma membrane contain porine proteins here and here to maintain the uptaking of nutrients from outside and transport of them to inside of the cell, or transportation of bacterial metabolites from the cytoplasm to the outside and for secretion of them. Very important part of the gram-negative cell wall, even gram-negative cell wall, is named lipopolysaccharide or LPS. Lipopolysaccharide also is named by the term endotoxin and by the term O antigen. Endotoxin and O antigen, they are two epithets characterizing the same substance that is named the lipopolysaccharide. So we have to recognize three names of this structure, of this molecule, distinctive for gram-negative cells. The gram-positive cells, lipopolysaccharide is absent. So first name is chemical lipopolysaccharide. Second, the name of the substance based on the toxicity, capacity to cause poisoning of the human organism. This is a endotoxic activity, endotoxin. And it means toxin that is situated inside of cell before release. And third name is O antigen. O antigen, this is an antigenic molecule typical for many gram-negative bacteria, and antibodies against O antigens may recognize in the laboratory testing O antigen of one bacteria separated from O antigen of other. As endotoxin, lipopolysaccharide is responsible for many of the features of diseases, because toxin, this is a poison causing disease. Endotoxin, for example, may cause such symptoms of disease as the fever, this is the increase in body temperature, tissue necrosis, destroying of tissues, and shock. Shock, this is a pathophysiological reaction that is based on the very quick and very intensive fall of the blood pressure. When pressure of the blood falls very quickly, shock is developed, and prolonged shock may cause the death of the patients. One of the reactions typical for reaction of endotoxin lipopolysaccharide is so-called Schwarzman reaction. Schwarzman reaction, that is also named disseminated intravascular coagulation. No, intravascular coagulation is formation of the blood clots. Schwarzman reaction follow the release of large amounts of the bacterial lipopolysaccharide into the bloodstream. One comment. What does it mean, endotoxin? Endotoxin is named endo because it's the integral part of the cell wall. It means from cell wall, lipopolysaccharide may be released only after breaking down of all the bacterial cell. Until cell survives, lipopolysaccharide of this bacterium cannot affect human cells. But if bacterium is lysed by the phagocytes or by the antibiotics used to treat the salmonella, salmonella typhus, typhoid fever. But in these cases, bacteria becomes broken and the toxins are released, and after that they becomes activated for bonding to the human molecules, causing their dysfunction or destroying. It means lipopolysaccharide is named endotoxin because may be released only after lysis of bacterial cell, and only after this lysis endotoxin may attack human cells. Lipopolysaccharide, this is a complex name demonstrating that molecule of the substance consists of three different elements. Lipo, it means internal part of the lipopolysaccharide consists of the lipid A, that consists of lipid acids. What these fatty acids are shown by what these black tails. This is lipid A. But even this lipid A, the toxic part, harmful for human cells. In the leaf bacterial cells, these fatty acids contact to the fatty acids of the phospholipids, this is second line of the molecules, and because they're hydrophobic, hide it from the water. Lipid A binds to the green, shown as the green, core polysaccharide. Core polysaccharide consists usually of 5 or some more sugar linked to the lipid A. Core polysaccharide this itself binds to the somatic O antigen. Somatic O antigen in our figure, this painting is shown as a polymer from one mannose, one rhamnose, biliose, and galactose. But here, 4 residues of the carbohydrates of sugars is shown. Really, somatic antigen of the majority of bacteria consists up to the 25 molecules of the sugars from 3. It means lipopolysaccharide. This is a complex molecule consisting of the somatic O antigen that is protruded to the outside of the cell. The second intermediate part is the core polysaccharide that binds to the lipid A that serves as the toxic portion of the lipopolysaccharide that is named endotoxin. The next structure is the capsule. Capsule lies outside the cell wall, covering the entire cell. What here you see 2 photographs. In the upper photograph, cell wall is shown as the gray covering of the single bacteria, couple bacteria, short chains of the bacteria. In the low photograph, you see 2 cells of the bacillus anthracis surrounded by the green, shown green, capsules. In majority of bacteria, capsule is the utmost protective membrane consisting of polysaccharides. Some exclusions are known. One of them, bacillus anthracis, that has a capsule of the polymerized D-glutamic acid. D-glutamic acid is the amino acid. It means capsule of the bacillus anthracis consists of the primitive protein. Properties of the capsule. Capsule is the determinant of virulence. Determinant is element. Virulence, this is the capacity of causing disease. So it means capsule may participate in the development of the disease because may limit the ability of the phagocytotic human cells to engulf bacteria. It means capsule in many bacteria serve as the anti-phagocytotic virulence factor. Second function. Capsule may play a role in the adherence of bacteria to the human tissues. This is also determinant of the virulence, because adhesins, structures of microbes responsible for attachment to the human cells, are involved in the development of the disease. Third properties. Anti-serum against the capsule, it means antibodies that specifically react to the capsular antigens of these antibodies can be used for specific identification of the bacteria in the laboratories. And fourth, capsule polysaccharides are used as the antigens in the certain vaccines. Antigens in the certain vaccines. Vaccines, you know, they're medical preparations consisting of the components of the microbes that usually inoculated to the healthy people to generate specific immunity of these people against these microbes. What polysaccharide capsules of some bacteria, for example, of Neisseria meningitidis, may be used and are rarely used as the vaccines to prevent this disease in humans. Flagellum. In this scheme, you see flagellum molecule of the brown color and the green color. Flagellum usually is mounted to the body of the bacteria by the rings. Here you see 1, 2, 3, 4 rings. L ring is integrated to the outer membrane of the cell wall of the gram-negative bacterium. P ring integrated to the peptidoglycan layer of the cell wall. The third is MS ring and C ring. These rings are integrated to the plasma membrane of the bacterial cell. Usually, one rotation of the flagellum is due to rotation of the rings, upper and lower, to the different directions. And as a result, flagellum makes the twisting movements that propel the cell, causing the movement. So, chemically, flagellum is the filament consists of the contractile molecule named flagellin. The energy for movement is the cell secures in the form of the so-called proton motive force. This proton motive force is shown in our scheme in the lower part of the flagellum when MS rings and C rings are integrated to the cell membrane of the bacterium. You know that cell membrane is involved in the synthesis of energy. What does it mean, energy, production of energy? First of all, this is a creating of the electric charge. What do you see here? You see that external part of the phospholipid layer of the plasma membrane accumulates positively charged ions, for example, hydrogen ions, on the external surface of the plasma membrane. In the same time, in the low part of the cell membrane, near to the low part, are accumulated ions with charged differently, with the negative charge. And what this difference of potential may be used by proteins surrounding MS ring and R ring of the flagellum to rotate flagellum. It means to rotate some of the proteins must be transported from outside to the inside of cell, and some of the negatively charged chlorine ions, for example, from the internal part to the external. It makes the proton motive force smaller, but for this excess of the protons, with transport of protons, gives a chance to flagellum to rotate. Usually, not only will this proton motive force serves as the preserved energy in the bacteria. In the cytoplasm occur molecules of the adenosine triphosphate. This is also energetic molecule. Energy of that may be used to rotate flagellum. Energy about MECU is the two forms, in the form of the adenosine triphosphate and in the form of the proton motive force on the membrane. This energy really is used for realizing and maintaining of all the functional cells, of the dividing, producing toxins, engulfment of the nutrients from cell, and for the motility. Here you see about flagellum. Patterns of the flagellal distribution. Different bacteria have the flagellum situated in different part of the bacterial cells. And this number of flagella and the location of flagella may be used in the identification of bacteria. Bacterium A, shown here, is named monotrichate or monotrichous bacterium. Mono, it means one. This bacterium has one flagellum and the one pole of the cell. B. If bacterium has not one but many beam of flagella situated at one pole of the bacterial cell, they are named lophotrichous bacteria or lophotrichate bacteria. Bacterium B. Bacterium C. You see that in the bacterium C, bacterium has two flagellum. One is situated in one pole of the cell, a second in the other pole of the cell. But this type of bacteria is named amphitrichous bacteria or amphitrichate. And bacterium D, the lowest bacterium, flagella spread over all the surface of bacteria. This bacterium is named peritrichous. It means monotrichous, one flagellum, peritrichous, many that surround all the surface of bacteria, lophotrichous, one beam of the flagella at one pole, and amphitrichous, two flagella, one at one pole, second at second pole. Bacterial endospores. Well, this is a very, very important thing. In the contrast to all other elements that we tried to explain before, endospores are not the organelles. What you see, for example, with one endospore, the bubbles, purple color, second, third, are the resting forms or resting stages of bacteria. Endospores, they are resting stage of bacteria that appear at a certain moment of the life cycle of the bacteria. And endospores of bacteria contain all the genome of this bacteria, contain about 10% of the cytoplasm of bacteria, and about 10% of the ribosomes of the bacteria. Usually presence of endospores is typical for two genera of medically important gram-positive bacteria, genus Clostridium and genus Bacillus. Formation of spores by the Clostridia bacilli is named by the term sporulation, sporulation, sporulation. Sporulation or spore formation helps bacteria to survive unfavorable environmental conditions. For example, conditions where nutrients disappear, or conditions where toxic substances accumulate in the medium where bacterium lives, or moderate heating. Against this heating, spore may protect bacterium. In this slide you see process of the formation of spore and germination of the spore. This is life cycle of the bacterium, of the Clostridium bacillus species. Vegetative cell. Vegetative means cell capable of replicating. When appears in the unfavorable conditions, form the cross wall and concentrate all the bacterial chromosome at the smaller part of the cytoplasm that is separated from other part of the surface by the cross wall that in principle is similar to the cross wall that is formed during the replication of bacteria. But in this small element separated by cross wall from other cell of the bacteria, other part of the bacterial cell, is accumulated genome and all the structural elements is required for surviving. Other parts of bacteria becomes degraded. Before this degradation, endospore acquires additional coats. Coats become very rigid, strong, and protective. Finally, the cytoplasm of the bacterium is destroyed, leaks, and in the growth medium appear free endospore. Why endo? Because the spore is formed inside of the bacterial cell that produces the spore. Spores are resistant to desiccation, radiation, most disinfectants, and heat. Spores of all medically important bacteria are destroyed, may be destroyed, by the manipulation named autoclaving. Autoclaving is a special device that in the working chamber may destroy spores of the bacteria, because in the working chamber of autoclave is created high pressure and high temperature. This is a method of the sterilization where steam under the pressure is used. It means steam is overheated and usually temperature required is more than 120 Celsius. And at this temperature, during 15 to 30 minutes, spores of the really all the non-medically important bacteria becomes killed. If you use only boiling at the 100 Celsius, this may be not enough to kill spores. Each bacterium forms only one spore. Due to this, sporulation of the bacteria is not considered as a replication of bacteria. Spore cannot replicate. Only vegetative cell may replicate. One vegetative cell may produce only one spore. After that, vegetative cell is destroyed. And the spore, the descendant of this vegetative cell, may waits for appearance of the better conditions in the future, and the future will germinate. Will germinate to the spore. What here in the photograph you see structure of spore. The spore is surrounded by a thing covering called the exosporium. What here this is a gray external covering of the bacterium. Exosporium surrounds second coat that is named spore coat. This is a more strong protecting layer of the spore. Spore coat lies beneath the exosporium, and is composed with proteins. These proteins is possible for spore resistance to chemicals compounds. Thick peptidoglycan cortex, CX, because beneath the spore coat. This peptidoglycan surrounds the spore core. Inside of the spore we can find DNA. DNA here is shown as the colorless white zones, and the dots. These dots are the ribosomes that's situated in the cytoplasm inside of the spores. Here you see germination of spore. The spore resistance may be mediated by dibicalinic acid. This is a very specific acid that is found only in the spore. The germination, it means growth of the vegetative cell from the spore, germination of dormant spore into the active vegetative cell is a complex process and may be triggered by many nutrients. The shape, size, and location of spore is typical from certain bacteria and may be used for identification of bacterial cells. So, the last part of the lecture will try to use to characterize physiology, not structure, but physiology of bacteria. In that slide, you see two photographs. In the left photograph, you see how a laborant prepares stick culture. Stick culture, this is a method of inoculation of the bacterial inoculum by the bacteriological loop with attached bacteria to the surface of loop. When serial sticking on the surface by this loop spreads bacteria on the surface of the solid medium. Here you see a petri dish with the pinkish red nutrient agar. This is solid medium, and process that is shown is preparing of the stick culture. In the right photograph, you see growth of Escherichia coli, or the colonies of Escherichia coli, on the surface of the mid peptone agar, also solid growth medium. Nutrition of bacteria. Let's say about nutrition. For growth and multiplication, bacteria require water, sources of carbon, sources of oxygen, hydrogen, nitrogen, sulfur, and organic salts. About 10 of the chemical elements are so-called macroelements that required in large amounts to the bacteria. They constitute over 95% of the cell dry weight. These substances are carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, calcium, magnesium, and iron. Bacteria require several micronutrients, exception for micronutrients that are required in very small amounts. Among them, zinc, nickel, copper, and some other. These substances can be a part of the enzymes and cofactors of the enzymes responsible for activity of these proteins. In addition, all organisms require sources of energy and electrons. On the basis of the nutrient requirements, all the microorganisms may be subdivided into 6 groups that are subdivided on the basis of the sources: carbon source, energy sources, and electron source. Concerning carbon source, all the microbes are subdivided to autotrophs and heterotrophs. Autotrophs are the microbes that can use CO2, carbon dioxide, as the sole or principal biosynthetic carbon source. It means CO2 becomes enough to produce many complex chemical compounds contain carbon. They are autotrophs. Heterotrophs, they need to use reduced preformed organic molecules from other organisms as the sources of the carbon. CO2 is not enough. Heterotrophs need sugars, lipids, proteins, and other complex molecules, organic complex molecules, to borrow the carbon. This is a carbon source. Autotrophs, heterotrophs. Second, energy source. Concerning energy source, bacteria subdivided into phototrophs and chemotrophs. Phototrophs may use as the energy source sunlight. Sunlight is enough for phototrophs. Other so-called chemotrophs, they need oxidation of organic or inorganic compounds, and the energy is borrowed from the chemical bridges of the complex chemical substances, organic and inorganic. And the last, electron source. The basis of electron source of the bacteria are subdivided to litotrophs and organotrophs. Litotrophs use reduced inorganic molecules to uptake electrons. Organotrophs need organic molecules as the source of the electrons. Physical requirements for growth are different, but what here you see three maybe most important requirements. First, temperature. Temperature for optimal growth of the most bacterial organisms is between 20 to from 20 to 40 Celsius. Bacteria that have the optimal conditions, this temperature, 20 to 40 Celsius, are named mesophilic bacteria, mesophils. They are major pathogenic microbes causing human diseases and animal diseases. Mesophilic, 20 to 40 Celsius. Other bacteria are named psychrophilic bacteria, psychrophilic, that grow better at the temperature below 20 Celsius. Only few species of pathogenic bacteria need temperature below 20 to grow intensively. And third type of bacteria is named thermophilic, thermophils. They grow best at the high temperature. High means from 55 to 80 Celsius. Second physical parameter, osmotic pressure. That depends on the concentration of salts or sugars. The growth rate is affected by the osmotic pressure of the medium. Most bacteria, medically important bacteria, are able to tolerate from 1 to 2% of salt, for example, sodium chloride. But some bacteria are named halophilic. Halophilic bacteria may survive at the 10 or 15% of the sodium chloride. And what among halophilic bacteria, one of the most important is Staphylococcus aureus. Staphylococcus causing inflammatory separative diseases of soft tissues in humans. Halophilic, resistant for high concentration of salts, it means for high osmotic pressure. And the third characteristic, pH. You know that pH is a characteristic of concentration of the hydrogen ions. High concentration is reflected by the low number, and low concentration of hydrogen ions is characterized by the larger number. For the growth of the majority of the pathogenic bacteria is near neutrally or slightly alkaline. It means majority of the pathogenic bacteria, medically important, they need pH from 7 to 7.6. 7 to 7.2 they're neutral, pHs 7.5 and higher, they're slightly alkaline. Well, these conditions are required for growth. The next physiological function, next after the nutrition, is the oxygen requirement or respiration. Concerning oxygen requirement or types of respiration, all the bacteria are divided into, in 4 large groups. First group is named obligate aerobes. Obligate aerobes can grow only in the presence of high concentration of oxygen, about 20 to 24% in the air. Example of the obligate aerobes are Vibrio cholera, causative agents of cholera, and Mycobacterium tuberculosis, causative agent of this pulmonary disease. Second group are named facultative anaerobes or simply facultatives. Facultative anaerobes can grow in the presence of oxygen and in the absence of oxygen. They utilize oxygen to generate energy by respiration, if it's present, if oxygen is present. But they can use the fermentation, this anaerobic respiration process, fermentation pathway, in the absence of the oxygen. You understand that these bacteria are most adapted for surviving in absolutely different conditions. Example of these bacteria are Escherichia coli and other members of the Enterobacteriaceae family. Salmonella typhi, Shigella dysentery, Klebsiella pneumonia, and many others. Third type of bacteria is named microaerophilic. Microaerophilic bacteria grow best in the presence of the low concentrated oxygen, not more than 1%, or lower than 1% in the gaseous system. Examples of microaerophils are Clostridium perfringens, that causes gas gangrene, and Helicobacter pylori, that causes gastric ulcer. And the last, obligate anaerobes. Obligate anaerobes can grow in the presence of oxygen because they lack enzymes named superoxide dismutase and catalase. These enzymes splits two types of the most toxic for bacteria oxygen-containing products. They are hydrogen peroxide and free superoxide radicals. Lack of the superoxide dismutase and catalase make these bacteria very sensitive to the oxygen that kills them. One word about aerobic organisms. How they use oxygen. Aerobes generate the energy, respiration, this is a process of generation of energy. Aerobes generate the energy only through oxidation or oxidative phosphorylation. This process is based on the use of the atmospheric oxygen, O2, as the ultimate hydrogen acceptor and the cytochromes. The process is named respiration. It means in the process of oxidation, oxygen is required and bacteria use oxygen as the ultimate hydrogen acceptor. It means in the plasma membrane, hydrogen is transmitted from one substance to other, and this transfer of the hydrogen from one to other helps in the accumulation of energy in the form of the adenosine phosphate or the electron motive force. What Altman's acceptor of this hydrogen is oxygen. So hydrogen is accepted together with electrons. It means acceptor of hydrogen, acceptor of electron is oxygen. The electron transport system of the bacteria, because transfer of the protons associated to transfer of the electrons, the electron transport system of bacteria is located on the cell membrane, at the depth of the cell membrane, and is composed of the cytochrome enzymes, for example, these cytochrome enzymes, flavoproteins, lipid cofactors such as vitamin K and coenzyme Q. Aerobic metabolism generates 38 adenosine phosphate molecules after oxidation of only one glucose molecule. What about we say? We say if during the nutrition one glucose molecule is transported through the porin channels inside of the bacterium, this glucose start to be cutted for the smaller molecules, and from the bridges broken in this process, electrons are borrowed and transferred to the other substances, electrons and hydrogen ions. With this transport of electron and hydrogen ions gives 38 androgen phosphate molecules after oxidative degradation of only one glucose molecule. This is a very high effectiveness of use of the neutron substrate. In the contrast, anaerobes use a so-called substrate level phosphorylation, not the oxidative phosphorylation, when oxygen is used for aeros, but substrate level phosphorylation. It means ultimate acceptor of the hydrogen would be not the oxygen, but the larger compound. Substrate level phosphorylation as the primary mean of the energy production is used by these bacteria, anaerobic bacteria. In the process of the substrate level phosphorylation of the glucose molecule and other carbohydrate molecules, pyruvic acid is produced from glucose. Process of destroying of glucose, the nutrient substance, gives from one glucose molecule two molecules of pyruvic acid. After that, pyruvic acid is converted to the lactic acid. And this conversion of pyruvic to the lactic acid maintains the circulation of the hydrogen. Do you remember we told about acceptor of hydrogen? This also hydrogen proton movement is important. It means what pyruvic acid transferring to lactic acid causes oxidizing of one reduced molecule of the nicotinamide dinucleotide protonated. When proton is removed from this molecule, this proton is transported from the pyruvic acid to the lactic acid. What you see here, two carbon atoms with a double chemical bridge to the oxygen. Lactic acid has only one. This compound, a second, becomes converted from oxygen to the OH hydroxyl radical. What circulation of the reduced and protonated forms of the nucleotide dinucleotide phosphide is the process of the degradation of glucose and converting glucose to the end products. This is a basis of the anaerobic respiration or substrate level phosphorylation. Not only lactic acid is synthesized in this way. Other end products of the process are acids, not lactic acid, for example, alcohols, gases, for example, H2S. All their characteristic end products of bacterial anaerobic substrate level phosphorylation. If glucose may be degraded in a certain bacteria, we say that bacterium is glucose positive. It means can ferment glucose. If we replace glucose by lactose, and lactose is involved in this process of transformation to the pyruvic acid and lactic acid, we say that this bacterium is lactose positive. It means may ferment lactose. What synthesis of the acid products in the process of the metabolism of the different carbohydrates is used in the identification of these carbohydrates, because different bacteria may ferment different carbohydrates, not all. And the spectrum of this substrate level phosphorylation is widely used in the biochemical identification of bacteria. No, the same process also is shown here. What do you see here? The anaerobes use substrate level phosphorylation or fermentation. What glucose is cut for two pyruvic acids because two electrons are uptaken by what glucose molecule, the process of its degradation. Two molecules of pyruvic acids after that are transformed to the two molecules of lactic acid. What is headed with the NAT H and NAT here? The process of the cleaving of the glucose into pyruvic acids, not positively charged two molecule NAT, nicotinamide dinucleotide, give two electrons for the glucose to maintain its cleaving to two molecules of pyruvic acid. After that, pyruvic acid uptakes two electrons in the process of the oxidation of NAT with the formation of the two molecules of lactose. Lactose, this is a typical anaerobic end product of the anaerobic metabolism of the glucose. Well, this way, anaerobic way, is faster than oxidative phosphorylation of the aerobic bacteria, but yields less energy. Two oxidative phosphate molecules only are formed from one glucose molecule, because end product in this metabolism would be lactic acid, not the CO2, but lactic acid. Bacteria division. Bacteria division is based on the accumulation of the increased amount of the cytoplasmic organelles in the maternal cell after the formation of cross wall with the appearance of two daughter molecules of the absolutely identical size and identical genome. The process is named binary fission, typical for bacteria. Before binary fission occurs, the cell must copy its DNA and segregate these DNA copies to opposite ends of the cell, maternal cell. Then the many types, about 10 types of proteins, start to be synthesized to form the cross-wall between two future daughter cells. One of the most important proteins synthesized, forming what this ring shown with blue, green here, is named protein FTSZ. This FTSZ forms the and assemble into the ring-like structure at the center of the cell. The septum is formed through the inward growth of the cytoplasmic membrane. You know that this inward synthesis of invaginates is named mesosomes. Other components of the division apparatus is assembled as the FTSZ ring. This machinery with this ring contracted, causing with formation of the cross wall and subdividing of the two halves of the maternal cell to the daughter cells. As division occurs, the cytoplasm is cleaved in two, and in many bacteria, new cells wall is synthesized. Bacteria grow in the laboratories usually occurs in three forms. First form of the bacterial growth, this is a growth on the surface of the solid growth medium, so-called agar medium. By the development of the colonies, what this from the growth shaded root of colonies. You see here many colors of different cells and color on the surface of the growth medium. Colonies are macroscopic, not micro, macroscopic product of from 20 to 30 cell divisions of a single cell. For example, if you see separate colony, it means that this bacterial population consists of descendants of one maternal cell that was involved in the from 20 to 30, maybe 40 cycles of the bacterial division. This is a colony. Second growth may be maintained in the broth liquid media. In the broth liquid media, after inoculation of bacteria to the growth media, after 20 hours, bacteria accumulate, producing turbidity. If you see turbidity, visible turbidity, it means that concentration of bacteria in this liquid is from 100 to 1,000 million bacterial cells per milliliter. But some bacteria elevate and form the pellicle on the surface of the growth media. Some bacteria sediment, forming pellets on the bottom of the tubes. Third type of the bacterial growth that may be maintained and studied in the laboratory is named biofilm formation. What does it mean, biofilm formation? When on the surface of the substrate, initially sediment proteins from the culture, these proteins form the layer, sticky layer on the surface of the substrate. Bacteria that float in the substrate sediment to this layer. Some of them sediment and absorb to this layer strongly and replicate here. Some bacteria detached, some accumulated in this area. When concentration of accumulated bacteria achieves the critically important concentration, these cells start to produce autoinducers. They are different for different bacteria. Autoinducers usually enhance replication of cells and stimulate synthesis by the cells of the substances, synthesis that was inhibited before. What some autoinducers, if you imagine that the surface of human cell, may make the cells absorb to the surface of the human tissue, start producing toxins that will destroy human tissues. Before, they were suppressed, genes that code for this toxin. And when the concentration of cell becomes enough high, they are activated and cells start to demonstrate their pathogenicity. In this moment, not only toxins are produced, bacteria start to produce external carbohydrates with a high concentration, so-called exopolymers. Usually these exopolymers are bacterial polysaccharides that form the layer that becomes more and more and more large and deep. Bacteria becomes integrated inside of the layer of these exopolymers. They progressively replicate, produce more number of the exopolymers. And finally, what matrix of the colony, consisting of the polysaccharide matrix and bacterial cells, becomes very large. Pieces of this large material becomes detached and float in the growth medium. What this polysaccharide layer sedimented on the substrate that contains inside bacteria producing bacterial products, including what is bacterial polysaccharides, what this complex of the bacteria and exopolymers is named biofilm. Biofilm help bacteria to survive in the environment, make them less resistant for antibiotics, for example. Some words about methods of bacterial culture. In principle, two methods of bacterial culture are known: batch culture, that is shown here, and continuous culture, shown here. What does it mean, batch culture? Batch culture, when bacteria are grown in the closed system, usually in the liquid medium, in the closed tube or flask, these bacteria, this type of growth, when do you put this tube to the incubator, maintains 37 Celsius, they demonstrate batch culture. What are the typical periods of the formation of batch culture in this volume of the nutrient? First period of the batch culture, this is a lag phase from 0 to 10 hours. What here you see figure demonstrating this dynamic of the accumulation of bacteria. Lag phase is demonstrated by the adaptation of bacteria to the nutrients in the growth media. They don't replicate, but they become larger and accumulate energy for active replication. After finishing of this adaptation, bacteria start to replicate exponentially, very intensively, in the logarithmic manner. And the second phase is named logarithmic phase. What usually in the logarithmic phase, bacteria demonstrate the typical classical morphology and stain. Then, when concentration of bacteria becomes high, and when concentration nutrients falls, and concentration of the toxic bacterial products is increased, conditions for bacterial growth becomes pure. And the third phase that follows the log phase is named stationary phase. Stationary phase is characterized by maintaining of the concentration of bacteria at one level. Some bacteria replicate, some bacteria die, and thirdly, number of living bacteria becomes unchanged. This is a stationary phase. Then, when conditions in the growth system becomes very, very pure due to the progressive disappearance of nutrients and accumulation of toxic products, death or logarithmic decline phase is registered. This is a phase of the declining of the death of bacteria, and majority of cells becomes killed, and only some low proportion of cells survives. Of course, bacteria that survive in these conditions usually have the changed morphology. When saying about spore-forming bacteria start to produce spores in the stationary phase, and maximum spores is synthesized in the period of the logarithmic decline, logarithmic death of cells, because you understand that spores and the spores of bacteria usually synthesized when conditions of surviving bacteria becomes pure. In the contrast, in the continuous culture, what is happening in this case? When bacteria are grown, the fresh medium flows into the fermenter continuously, and part of the medium in the reactor is withdrawn from the fermenter at the same flow rate of the inlet flow. It means in the continuous culture, concentration of the nutrients is maintained at enough high level, and due to the removing of some of the old growth medium, toxic products are removed from the system, and bacteria may achieve plateau for this situation where the concentration is high, and this plateau may be maintained artificially unlimited time. It means in the continuous culture, lag phase is present, logarithmic phase is present, and plateau phase, stationary phase, without declining phase. Here we see 4 elements of the system for maintaining continuous cultures. In the left part of the photograph, you see bottle with a fresh growth medium. Then you see pump that delivers the growth medium to the reactor, or this is a third element reactor, where bacteria occur, metabolize. Some portions of this growth medium is removed and replaced by the fresh medium to maintain the concentration of bacteria at the maximum level. and in the right part you see control tower. This is a system that controls the correct maintenance of the regimen and the culture of the bacteria. Isolation of bacteria in the laboratory usually is based on the inoculation of bacteria, calcium bacteria, to the surface of growth medium in the petri dish, by the stabbing of the bacteria by the inoculation loop to the depth of the agar deep tube, by the inoculation of bacteria to the broth liquid medium, or by the inoculation of bacteria at the surface of the agar slant. In this photograph you see method of the inoculation of bacteria at the surface of agar that is named streak culture. Streak culture, this is a creating of the sets of streaks by the inoculation loop that before was contaminated by bacteria that we want to grow on the surface of this agar. Of course, all the manipulations must be done near to the flame burner, because heating of the air near to the place where you manipulate with petri dish creates the elevating flow of the air, and bacteria don't sediment to the surface of the agar from the environment, and this manipulation prevents the inoculation to the growth medium of the parasitic bacteria that are absent in the patient's specimen but present in the air of the laboratory. After preparing of the stick culture, bacteria cultured in the incubator at the 37 Celsius for 20 hours about. During this time, bacteria form colonies. You take the isolated colony. Why isolated? Because only isolated colony may consist of the bacterial generation, bacterial mass consisting of the progeny of only one bacterial cell. To keep the so-called pure culture, you have to take the colony isolated, thinking that all the bacteria that will appear here are identical because descendants of only one cell that was inoculated here before. So if do you see culture here, isolated colony, you wipe part of this colony by the inoculation loop and inoculate bacteria to the surface of the agar slant. This subculture is named stroke culture, the second step of isolation of pure culture of the bacterial colony. In this scheme you see simplified scheme of the identification of bacteria. No, let's imagine that we take gram-positive bacteria, gram-positive algorithm. First method that we use for culturing is named microscopy. Now, for example, we make gram-stained bacteria and we'll see either rods, either cocci. Rods, we wouldn't discuss this how they identified. Let's see what we can do with cocci. Cocci brown bacteria. This cocci may be undergone to the additional test for presence of enzyme named catalase. Catalase is an enzyme that cleaves hydrogen peroxide. Cocci, on the basis of this test, may be subdivided to catalase positive and catalase negative. Positive, the bacteria that contain catalase negative not. Staphylococcus usually are catalase positive, streptococcus usually are catalase negative. Staphylococci, after that, are examined for the presence of the second enzyme that is named coagulase. Coagulase may coagulate blood plasma of donors and animals. Staphylococci that are coagulase positive, it means have this enzyme, are named Staphylococcus aureus, identification for species. Coagulase negative staphylococci may be different. For example, Staphylococcus epidermidis, saprophyticus, and so on. Well, this is one of the way of the identification of the bacteria. In principle, methods of the identification of bacteria are based on the microscopic morphology examination, what you see here, Strepta bacilli. Then, method of macroscopic morphology identification, where do you examine colonies, size, transparency, and other properties of colonies. Then you may examine isolated bacteria by biochemical tests. You see 1, 2, 3, 4, 5 tubes. All these tubes contain the same growth medium that has indicators of pH and hydrogen sulfate synthesis. First tube demonstrates slant of the triple sugar ion agar before inoculation of bacteria. Second, this is a result of inoculation to the triple sugar ion agar of the Salmonella typhimurium that produces very large amounts of the hydrogen sulfate. As a result, agar becomes black. Third tube demonstrates very intensive fermentation of the lactose by the bacterium. Hydrogen sulfate is not synthesized, but color of all the elements of the agar becomes yellow, not pink, but yellow. It means that lactic acid is formed. It usually in this medium occurs lactic no, lactose positive bacteria, lactic acid. The fourth tube demonstrates that lactic acid accumulates differently in different parts of this growth medium, and in the fifth part you see moderate, not very high, but accumulation of hydrogen sulfate. One of the methods of identification of bacteria is named biotyping, using animals, laboratory animals that after inoculation by the bacteria may die or survive depending on the change the conditions of the inoculating of bacteria and the maintaining of these animals in laboratory. Serotyping method, we would describe this because this is a method directed for finding of the antibodies against all antigens of bacteria. Phage typing is widely used. Bacteriophages are viruses that destroy the certain types of the bacteria. They're highly specific, these bacteriophages, for the bacteria. If you culture bacteria in the solid medium, this is the culture of Bacillus anthracis. Specific Bacillus anthracis bacteriophage, after dropping to the bacterial culture, demonstrates lysis of bacteria. If specific phage may lyse this bacteria, we say that this culture is Bacillus anthracis because it's sensitive for bacteriophage. And genotype testing is widely now used to identify bacteria. Here you see a slide with the different morphological characteristics of bacteria. Here, different macroscopic characteristic of colonies: red pigmented, yellow pigmented, flat surface of bacteria, what rhizoid surface of bacteria, another flat, what filamentous margins of the not bacteria, of the bacterial culture of the colonies. Here you see different variants of the colonies that may be evaluated in the form, for example, of colonies. Some of them may be punctiform, some circular, some filamentous, irregular, rhizoid. Rhizoid, it means forming branched elements that looks like the root. Spindle or lens-shaped colonies. Concerning elevation, bacterial colonies may be flat, raised, convex, pulvinate, concave, ambonate, and other. We discussed this experiment before. How one growth medium contain three different types of sugars may differently react to the different bacteria that have different fermentative characteristics. Here you see this diffusion test demonstrating susceptibility of bacterium to the antibiotics. Bacteria inoculated to all the surface of petri dish by the capillary culture. After that, 1, 2, 3, 4, 5, 6, 7 discs soaked with 7 different antibacterial drugs are placed on the surface. After 20 hours of the culturing, diffusing of the antibiotics to the growth medium either prevent growth of bacteria near to the disc, as is shown here and here, either don't prevent, that is shown here. This test demonstrates what is the susceptibility of bacteria to the antibiotics. And finally, pay attention to the genotypic test. Here you see result of the electrophoresis, electrophoretic distribution of the oligonucleotides that were produced by the contact of the DNA of microorganisms to the certain restriction enzymes, and different structure of the sequences of nucleotides give the different bands that have different molecular weight. This is one of the methods of the genotyping of the bacterial and now human cells also. Now this is the finish of my lecture. Thank you for your attention. Goodbye. See you in the next lecture. Goodbye. Дмитрий, я уже только что закончил чтение лекции. Что я теперь должен сделать? Так, как я могу это сделать? Я должен вот... Так, подождите. Вот у меня есть... вот сама презентация, да? Вот нажимаю красный крестик. Что-что? Сейчас смотрю. Выход. Это вот там, где вот управление, остановить трансляцию, начать запись, да? Вот там